All right, so you've been sending us mountains of research, really wanting to wrap your heads around those crucial steps it takes to bring a new medicine to the people who desperately need it. Taking it from the lab to the pharmacy, the whole 9 yards.
It's quite the journey, isn't it?
Oh, absolutely. And today, we're zeroing in on a pivotal moment in that journey, the successful phase 3 clinical trial.
Ah, yes, phase three, a real proving ground, you could say. It's where a drug that's shown some early promise in those smaller studies, phase one and two, finally gets put to the test in a much larger and more diverse group of patients.
Exactly. And, we've been poring over all the material you sent, clinical trial handbooks, drug development guides, even diving into those dense regulatory documents like the CFR.
Not to mention those fascinating case studies from medicinal chemistry and pharmacokinetics really paints a picture of what goes on behind the scenes.
It really does. So for this deep dive, let's really try to nail down what makes a phase 3 trial successful. Walk through the design, the execution, those inevitable challenges that pop up, and of course, how they ultimately demonstrate that statistical significance that everyone's looking for.
Sounds like a plan. Because let's face it, phase three is where the rubber meets the road. It's the evidence that regulatory agencies scrutinize before they even consider giving a thumbs up to a new drug.
And, for our listeners out there, this is the stuff that impacts whether a potentially life-changing treatment makes it to market or not. It's a big deal.
Couldn't agree more. So, where should we start?
Well, I think it makes sense to begin right at the beginning with the trial design. Seems like that sets the stage for everything that follows.
Absolutely, the design is the foundation. And as one of your sources rightly pointed out, there's this huge emphasis on tailoring the design and the methods you'll use to analyze the data to the specific research question you're asking and the unique characteristics of the patient group you're studying. It's not a cookie-cutter approach by any means. They really stress the importance of considering different statistical strategies, too, to make sure you're using the most appropriate ones.
Right, so it's not just one size fits all. A trial for a new heart medication would likely look quite different from one testing a novel cancer therapy.
Exactly. Different diseases, different patient populations, different endpoints, all these factors demand a bespoke approach to the trial design.
Now, something that struck me was the need to keep the hypothesis clear and streamlined, especially when you're dealing with smaller trials.
Oh, absolutely. A clear, focused hypothesis is like a beacon guiding the entire trial and making the analysis much cleaner.
So you don't get lost in the weeds. And speaking of crucial design elements, there's the sample size calculation. You got to make sure you enroll enough patients to actually see a real difference between the treatment groups.
Yeah, you hit the nail on the head. It's all about statistical power, having enough participants to detect a real effect if it truly exists. If your trial is underpowered, you risk missing a potentially effective treatment. And that's a huge disservice to patients who are waiting for new options.
So how do you go about figuring out that magic number, the right sample size?
Well, it's not magic, but it definitely involves some statistical wizardry. Several factors come into play. First, you need to estimate the expected size of the treatment effect. How much better do you anticipate the new drug will perform compared to, say, a placebo or the current standard treatment? Then there's the level of significance, which basically sets the threshold for how much risk you're willing to accept of concluding there's a difference when there really isn't one.
Sort of like a safety net to avoid jumping to conclusions.
Precisely. And then, of course, there's the power, which, as we said, is the trial's ability to detect a true effect if it's there. It's a delicate balancing act between these different considerations.
Makes sense. Now, I came across these terms, intention to treat and per protocol analysis. What's the distinction there and why are both important?
Great question. These are two primary approaches to analyzing the data once the trial is wrapped up. Intention to treat, or ITT, means you analyze the data for everyone who was initially randomized into the different treatment groups, regardless of whether they perfectly adhered to the treatment plan or even finished the trial. Per protocol analysis, on the other hand, focuses specifically on the subset of patients who stuck to the protocol precisely as it was laid out.
So ITT takes a broader view, including those who might have veered off course a bit.
Exactly. It reflects real-world scenarios where patients don't always take their medication exactly as prescribed or might discontinue treatment for various reasons. By including everyone who was randomized, ITT helps mitigate potential biases that could arise if, say, people who experienced side effects and dropped out were systematically different between those getting the new drug and those on the control arm. It provides a more conservative and arguably more clinically relevant picture.
More like what you'd see in everyday practice.
Precisely. Per protocol analysis, though, offers a clearer picture of how the drug performs under ideal circumstances, when patients adhere perfectly. It can be useful for understanding the drug's full potential, but it might not reflect the complexities of real-world use as accurately.
I see. So it's like looking at both the ideal scenario and the real-world scenario to get a complete understanding.
Right, and choosing the appropriate analysis method for the specific study is critical. You don't want to be switching horses midstream or picking the one that gives you the most favorable result. You determine this upfront, based on the study design, your objectives, and what outcomes you're measuring. It's all laid out in the statistical analysis plan.
Transparency is key. Now, what about these surrogate endpoints I keep seeing? They seem to be a bit of a shortcut.
They can be, in a sense. Surrogate endpoints are essentially measurable stand-ins for the real clinical outcomes we're ultimately interested in. Think of things like changes in blood pressure or cholesterol levels, tumor shrinkage things that are thought to be good predictors of, say, a reduced risk of heart attack or stroke or slower disease progression.
So instead of waiting years to see if the drug actually prevents heart attacks, you might look at whether it lowers blood pressure within a shorter time frame.
Exactly. They can be particularly useful in those earlier phase 2 trials to get a quicker read on whether a drug is showing promise. It can potentially speed up the development process and save resources. But,
There's always a but.
right, while surrogate endpoints can provide valuable early insights, they don't always perfectly reflect the actual clinical benefit. For a drug to get the regulatory green light, you typically need those definitive phase 3 trials that show a clear impact on how patients feel, function, or survive, those clinically meaningful outcomes.
So surrogates can be a helpful stepping stone, but the ultimate proof lies in those real-world benefits.
Absolutely. And sometimes that link between the surrogate and the clinical benefit isn't as strong as we initially thought, which can be a big setback.
Now, this concept of non-inferiority trials, the one always throws me for a loop. It seems counterintuitive. Wouldn't you always want to show that a new drug is better than what's already available?
It would seem that way, but it's not always the primary goal. In a traditional superiority trial, you're aiming to demonstrate that the new treatment is statistically and clinically superior to a placebo or the existing gold standard. But in a non-inferiority trial, the objective is to show that the new treatment is at least as good as the established therapy within a predefined margin.
So proving it's not worse, but not necessarily proving it's better, when does that approach come into play?
It's often used when there's already an effective treatment out there, but the new drug might offer other advantages, perhaps fewer side effects, a more convenient dosing schedule, or a different way of working in the body without compromising on efficacy.
So maybe it's gentler on the stomach or taken once a day instead of twice a day. Things that could improve a patient's quality of life.
Exactly. It's all about finding treatments that offer a better overall experience for patients, even if they don't necessarily deliver a huge leap in efficacy.
But how does that change how you analyze the data?
Well, in a superiority trial, you're typically looking at whether the confidence interval, that range of plausible values for the difference in treatment effects excludes zero. But in a non-inferiority trial, you're focused on whether the lower bound of that confidence interval stays above a predetermined non-inferiority margin. Basically, it's the maximum amount by which the new treatment could be less effective than the standard while still being considered clinically acceptable.
So there's a bit of wiggle room, but it's carefully defined.
Precisely. And here's another interesting twist. A large p-value in a non-inferiority trial, unlike in a superiority trial, doesn't necessarily mean the two treatments are equivalent. It just means you haven't conclusively proven that the new treatment is worse than the standard by more than that preset margin. It's subtle, but important.
Definitely a lot of nuances to keep straight. Okay, last question on trial design. I saw something about stratification by treatment center. Why would you do that, and what implications does it have for the analysis?
Stratification is a clever technique used to account for potential variations that might exist across different clinical sites participating in the trial. For instance, patient populations might differ slightly between hospitals, or there might be subtle differences in standard medical practices.
Right, because a large multi-center trial could involve hospitals in different regions or even different countries.
Exactly. By stratifying, basically creating subgroups based on the treatment center, and then ensuring that the treatment groups are balanced within each center, you can reduce the risk of these site-specific differences, skewing the overall treatment effect. But remember, if you stratify the design, you need to account for that in the analysis too.
Got it. You've got to stay consistent. Okay, so we've covered the importance of a rock-solid trial design. But even with the best blueprint, things can go awry during the execution of a large Phase 3 trial. What are some of the challenges that researchers face, and how do they ensure the quality and integrity of the trial?
You're absolutely right. Execution is everything. Even with a brilliant design, a trial can falter if it's not managed carefully. And this is where good clinical practice, or GCP, comes in. You'll see this acronym a lot. It's essentially the gold standard for conducting ethical and scientifically sound clinical trials with the globally accepted guidelines outlined in ICH E6R2.
So it's a framework for doing things the right way, both ethically and scientifically.
Precisely. GCP ensures that the rights, safety, and well-being of those participating in the trial are protected, and it also ensures that the data collected is credible and reliable.
Makes sense. When you're dealing with human subjects, ethical considerations have to be front and center. How are those safeguards put into practice?
One of the cornerstones of GCP is the role of institutional review boards, or IRBs. These are independent committees made up of medical professionals, scientists, and even members of the community. Their job is to review and approve the trial protocol and all related study documents before the trial can even begin.
So they're like an independent ethics watchdog.
You could say that. Their primary concern is ensuring that the potential benefits of the research outweigh any risks to the participants, and that there are appropriate measures in place to protect their rights and welfare. It's a huge responsibility, and it underscores how seriously we take the ethical dimensions of clinical research.
Absolutely. And of course, there's the critical process of getting informed consent from each participant.
Oh, informed consent is non-negotiable. It's the bedrock of ethical research. Before anyone can enroll in a trial, they have to be given comprehensive information about the study, its purpose, the procedures involved, the potential risks and benefits, what alternative treatments might be available, and their rights as a participant, including the right to withdraw from the trial at any time, no questions asked. And this information has to be presented in a clear and understandable way, not buried in jargon. Then, if they agree to participate, they sign a consent form.
So it's not just a formality, it's a genuine process of ensuring people understand what they're getting into.
Exactly, and it's an ongoing process throughout the trial. If any new information comes up that might affect their decision, they have to be informed promptly.
Transparency is key. Now, I can only imagine the sheer volume of data generated by a Phase 3 trial. How do you ensure that all that information is accurate and reliable?
You're right, we're talking about massive amounts of data. That's why robust data management and quality control are absolutely essential throughout the entire trial process. This includes having clear, standardized procedures for collecting the data, entering it into databases, and storing it securely. There also need to be regular checks and audits to catch any errors, inconsistencies, or missing data. Maintaining the integrity and reliability of the data from the moment it's collected to the final analysis is crucial for the credibility of the trial's findings.
I also saw mention of clinical research organizations, or CROs. What role do they typically play in these large Phase 3 trials?
CROs are like specialized partners that provide a wide range of support services to pharmaceutical and biotech companies conducting clinical trials. Think of them as extensions of the research team. Sponsors, especially smaller companies, often outsource certain aspects of their trials to CROs. Things like trial management, monitoring the clinical sites, data management, even the statistical analysis and regulatory affairs.
So they bring expertise in areas where the sponsor might not have the in-house resources or experience.
Exactly. It allows sponsors to tap into the specialized skills and infrastructure of CROs without having to build everything from scratch. Similarly, there are other contract service organizations, or CSOs, that also play various roles in executing clinical studies.
Makes sense. Now, let's be realistic. In a complex undertaking like a Phase 3 trial, it's almost inevitable that some changes or adjustments might be needed along the way. How are those handled?
Right. You can plan meticulously, but there's always the possibility of unforeseen circumstances or the need for mid-course corrections. If substantial changes to the study protocol become necessary after the trial has already begun, the sponsor typically has to submit a formal protocol amendment to the regulatory agency, like the FDA in the US.
So you can't just make changes on the fly.
Absolutely not. You need regulatory oversight to ensure that any changes don't compromise patient safety or the scientific validity of the trial. This could involve things like adding a new group of patients, modifying the drug dosage, or even adding new endpoints to measure. Even for less dramatic changes, or to start a new study that wasn't originally described in the investigational new drug application, you usually need a protocol amendment.
It's all about maintaining that rigor and transparency. Now, this last point on execution might seem a bit surprising, but it highlights the importance of something as seemingly mundane as equipment design.
It might seem trivial, but it's not. The design, size, placement of the equipment used in the trial, whether it's for administering the drug, monitoring vital signs, or processing samples, all of it can impact the smooth operation of the study. Equipment that's designed well makes things easier for the staff, simplifies cleaning and maintenance, and ultimately contributes to the quality and reliability of the data.
So even the seemingly small details matter.
Every detail matters. It all ties back to those overarching principles of good manufacturing practice, or GMP, which in a broader sense are all about ensuring the quality and integrity of the investigational product and the data generated around its use.
That makes sense.
Yeah.
Okay, so we've laid the groundwork with a solid design and ensured that the trial is executed with the highest quality and ethical standards. But the ultimate question is, does the drug actually work? How do researchers go about proving the statistical significance everyone's talking about?
All right, well, now we're getting to the heart of the matter, proving the drug's worth. And this is where the concept of the p-value takes center stage. You'll hear this term thrown around a lot, and for good reason. It's a fundamental concept in statistical hypothesis testing.
Okay, break it down for us. What exactly is a p-value?
In simple terms, the p-value is the probability of observing the results you got in your trial, or results even more extreme if there was actually no real difference between the treatment groups. In other words, it's the chance that the observed effect is just a fluke, a random occurrence, and not due to the drug itself.
So a tiny p-value is a good thing, right?
Exactly. A small p-value, typically less than 0.05, is what researchers are hoping for. It means that the observed effect is highly unlikely to have happened by chance alone if the drug had no real effect. It gives you more confidence to say, hey, this drug is actually doing something.
But I recall reading that the p-value, while important, isn't the be-all and end-all. There was something about confidence intervals too, right?
Yeah, you're right on the money. While the p-value tells you whether the effect is statistically significant, meaning it's unlikely due to random chance the confidence interval, or CI gives you a range of plausible values for the true treatment effect. For example, a 95% confidence interval means that if you were to repeat the study many times, you'd expect 95% of those calculated intervals to contain the true difference between the treatment groups.
So it gives you a sense of how precise your estimate of the treatment effect is.
Exactly. A narrow confidence interval indicates a more precise estimate, while a wider interval suggests more uncertainty. Even if two studies have the same p-value, the one with the narrower confidence interval might be considered more reliable.
So it's not just about statistical significance, it's also about the magnitude and precision of the effect.
Precisely. And remember our discussion about non-inferiority trials. The confidence interval plays a crucial role there too, because you're looking at whether the lower end of that interval stays above that predefined non-inferiority margin. It adds another layer of complexity to the interpretation.
It's all starting to come together. Now, phase 3 trials often collect a lot more data than just the primary efficacy endpoint, right? What about things like patient-reported outcomes, like their quality of life?
Oh, absolutely. Things like health-related quality of life, or HRQL, are often measured at multiple points throughout the trial. Analyzing this longitudinal data is important for understanding how the treatment affects patients' overall well-being over time.
So you're not just looking at whether the drug shrinks a tumor, you're also looking at how it impacts the patient's day-to-day life.
Exactly. It helps you see how the treatment effects evolve, whether any improvements are sustained, and also how things might differ for patients who drop out of the trial early.
Because if people who are feeling worse are more likely to withdraw, that could skew the results, couldn't it?
You got it. That's why sophisticated longitudinal analyses are important to tease out these potential biases and provide a more accurate picture of the treatment's true impact over time.
Now, I briefly came across the term adaptive designs. Can you shed some light on those? They sound pretty high-tech.
They are cutting edge. Adaptive designs basically allow for pre-planned modifications to the trial's design or statistical analysis based on the data that's accumulating as the trial's ongoing. It's like making course corrections in real time.
So you might adjust the sample size or tweak the treatment arms based on early signals from the data.
Exactly. This flexibility can potentially make trials more efficient and increase the odds of success, but it's not a free-for-all. Adaptive designs require careful planning and sophisticated statistical methods to make sure the results are still valid and reliable. It's a complex area, for sure.
Sounds like it. Okay, let's fast forward to the finish line. The phase 3 trial is a success. The drug hits its endpoints. The safety profile looks good. What happens next? Does the drug just magically appear on pharmacy shelves?
If only it were that simple. A successful phase 3 trial is a huge milestone, but it's just one step in the long and winding road to drone approval. The next major hurdle is submitting a new drug application, or NDA, to the FDA in the US, or a similar marketing authorization application to other regulatory agencies around the world.
So the NDA is like the big final exam where all the evidence is presented for scrutiny.
That's a great analogy. The NDA is a comprehensive package that includes all the data from preclinical studies, all phases of clinical trials, manufacturing information, quality control measures, labeling, everything.
It's got to be a mountain of paperwork.
It is, and one of the key things the FDA looks at is whether the company can consistently manufacture the drug at the required quality and on a large scale. It's not enough to produce a few good batches for the clinical trials. They need to demonstrate that they can reliably produce the drug for the masses.
Right, because consistency and quality are paramount for patient safety.
Absolutely. And the monitoring doesn't start there. Even after a drug is approved and hits the market, the work continues. This is where pharmacovigilance comes in. It's all about monitoring the drug's safety and effectiveness in the real world, where it's being used by a much larger and more diverse population.
So it's about keeping a watchful eye out for any potential safety issues that might not have shown up in the clinical trials.
Exactly. Doctors, patients, drug manufacturers, everyone is encouraged to report any suspected side effects or other safety concerns. The FDA has a system called MedWatch, specifically for this purpose, and companies are also required to promptly report any serious and unexpected side effects that come to their attention. And periodically, they have to submit safety update reports summarizing everything that's been learned about the drug's safety since it went on the market.
So it's a continuous process of evaluating and refining our understanding of the drug's safety profile.
Absolutely. And there are even proactive surveillance programs like the FDA's Sentinel initiative that use electronic health records and other large databases to actively look for potential safety signals in real-world clinical practice.
Wow, it's really a long-term commitment to ensuring that medications are both safe and effective. So to sum it all up, a successful phase 3 trial is a monumental achievement, but it's just one critical step in the long and complex journey of bringing a new medicine to patients. And that journey doesn't end with FDA approval. It continues through ongoing monitoring and evaluation in the real world.
We've covered a lot of ground today, but it's clear that the design, execution, and demonstration of statistical significance in phase 3 trials are absolutely essential for bringing new therapies to those who need them. And overcoming the inevitable challenges while always adhering to the highest ethical and quality standards is not optional. It's the only way to ensure that these trials succeed and ultimately benefit patients.
We've really taken a deep dive into the intricate workings of a successful phase 3 trial, giving you a much clearer picture of this crucial phase of drug development. But here's something to ponder as you continue to explore this fascinating field. Given all the factors we've discussed, the meticulous design, the complexities of execution, the statistical hurdles, what do you think is the single most crucial element that determines whether a phase 3 trial ultimately succeeds? And looking ahead, how might emerging technologies, perhaps like AI and machine learning, reshape this critical phase of drug development in the years to come?
It's a lot to think about, and we encourage you to keep digging, keep asking questions, and keep learning. Absolutely, and as always, we're here to help guide you on your journey. So keep those research requests coming.
We'll be back next week with another deep dive into the world of medicine and science. Until then, stay curious.